is Tiffany Aguirre, and I am a graduate student in the psychology department in Dr. Prati's lab, and this project is investigating sex differences and the motivational consequences of opioid withdrawal using a conditioned place aversion paradigm in rats. So, previous literature has shown that women experience faster escalation, higher cravings, and higher relapse rates of opioids. Despite this, there are still a lack of studies on sex differences in opioid withdrawal and its motivational consequences. So, that's exactly what we set out to do using a conditioned place aversion paradigm. So on day 1, we allowed rats to freely explore both chambers in order to establish no sense of biases. Then on day 2, animals were injected with saline and then locked in one chamber for 22 minutes in order to associate this chamber with feelings of neutrality, right? So they're not undergoing withdrawal here. On day 3, the next day, we injected them with morphine and then 4 hours later naloxone to precipitate withdrawal and then locked them in the other chamber so they associate this chamber with feelings of withdrawal. And then the next day, repeat what the last 2 days were. So inject with saline and then lock in the first chamber to associate that chamber with feelings of neutrality. And then the next day, inject with morphine and then 4 hours later naloxone and then lock them in the other chamber so they associate this with feelings of withdrawal. So then on the 6th day, which is our test day, we allow them to roam freely in both chambers and record how much time they spent in each chamber. So as we can see from our results, there were no sex differences in aversion. So in the behavior that was displayed, both males and females spent similar amounts of time avoiding the chamber that they associated with naloxone and withdrawal. So that was kind of a bummer. But then we came across the theory of convergent sex differences that states that behaviors elicited by both sexes could be similar, however, the underlying biological processes may be different. So this is why we continued the project and stained for CFOS, which is a biomarker that indicates neuronal activity. So we stained for CFOS in various brain areas and actually found significant sex differences in the infralimbic and prelimbic cortex, as well as the nucleus accumbens core and shell and the ventral tegmental area. We saw that males had significantly more CFOS expression in the infralimbic, prelimbic cortex, and the NAC core and shell than the females. So then we did the same procedure on saline animals in order to establish a control and still found that males had significant CFOS expression than their saline counterparts, indicating that these areas are being activated during aversion. What threw us in for a bit of a loop was that females had significantly more CFOS expression than males in the VTA as well as their female saline counterparts. So, needless to say, this was actually very exciting to find evidence for convergent sex differences in that we see that there is an activation in the infralimbic cortex, which regulates fear conditioning, and projects to the NAC shell, which regulates the effective processing of reward, as well as an activation in the prelimbic cortex, which is important for higher function cognitive abilities like decision making, and projects to the NAC core, which regulates motor processing of reward like drug seeking behaviors, suggesting that these two pathways moderate aversion in males. But the real exciting part was that in these same areas, there was no difference between treated females and their saline counterparts, suggesting that these areas weren't as active, and yet we still see this aversive behavior being displayed by females. So maybe, perhaps, there is a different pathway that moderates aversion in females. Thank you.